Okay, now this has to do with the diagrammatic representations of what I spoke to just now. So this is the glycolytic pathway where glucose gets converted to pyruvate. Right, this is the first diagram, and next shows the erythrocytes and the reactions when converting to pyruvate where there's no mitochondria, right, and lactate of cells with mitochondria. All right, and this is the formula of pyruvate being converted to lactate, right? And this is the effective cycle that we just spoke of, the conversion of glucose in its various stages. See, glucose, that's a glucose 6-phosphate, fructose 6-phosphate, right? And dehydrogenase acetone, dihydroxyacetone phosphate, and the glyceraldehyde 3-phosphate, right? And fructose 1,6 biphosphate, and then getting converted to glyceraldehyde 3-phosphate, and then to 1,3 biphosphoglycerate, right? Can you see that? And then 2-phosphoglycerate and phospho in all pyruvate, that too, finally to pyruvate. And see this list? These are the list of enzymes which I spoke about, which is that hexokinase, hexokinase, glucokinase in liver and pancreatic B cells, the phosphoglucose isomerase, 6-phosphofructose 1 kinase, or aldolase, triose phosphate isomerase, glyceraldehyde 3-phosphate dehydrogenase, phosphoglycerate kinase, phosphoglycerate mutase, enolase, and pyruvate kinase. All right. Next, the individual reactions. This is the first one of hexokinase and glucose, converting glucose to glucose 6-phosphate, right? You see that? Right. Next would be the action of phosphoglucose isomerase, which converts glucose 6-phosphate to fructose 6-phosphate, right? And the next would be fructose 6-phosphate, in the presence of 6-phosphofructose 1 kinase or PFK1 and ATP to form fructose 1,6 biphosphate. And then aldolase reaction, where you have fructose diphosphate 1,6 biphosphate getting converted to dihydroxyacetone phosphate and glyceraldehyde phosphate 3-phosphate. And then you have the action of dihydroxyacetone phosphate in the presence of triose phosphate isomerase to glyceraldehyde 3-phosphate. And the next one would be glyceraldehyde 3-phosphate in the presence of glyceraldehyde 3-phosphate dehydrogenase getting converted to 1,3 biphosphoglycerate. And the next one, 1,3 biphosphoglycerate in the presence of phosphoglycerate kinase and ADP forming an ATP giving 3-phosphoglycerate. And the number 8 would be 3-phosphoglycerate in the presence of phosphoglycerate mutase to phosphoglycerate. Then 2-phosphoglycerate, the presence of enolase liberating water to give phosphoenolpyruvate, and phosphoenolpyruvate in the presence of pyruvate kinase giving pyruvate, right? And this is the pyruvate dehydrogenase in presence of lactate dehydrogenase. When it gives lactate, it's anaerobic. This is the synthesis of 2,3 biphosphoglycerate, right? This is about the control of glycolysis, the graph here. If any of you need any of these diagrams as a screenshot, or as you want me to share the details with you, do let me know. connect me on the WhatsApp number I've told you, and I'll send it across. This is the picture of the hormonal control of fructose 2,6-biphosphonate in the liver. Right? These are important for the exams. This is about the synthesis of fructose 2,6-biphosphate. Okay, and the CN-dependent protein kinase action of phosphorylation causes an activation of the active site responsible for the synthesis of fructose 2,6-biphosphate and activation of the site responsible for the hydrolysis. Right? And then that's it. Should be fine with the details there, and I'll send it across. Put that another video. Could be a